Okay, the P53 gene, the tumor suppressor gene, functions as a repairman for the DNA, and also it goes around checking to see if a cell is needed or not, or if that cell cycle is needed or not, and if it's not, it stops it, or if the cell is defective, it will kill the cell, send it off to apoptosis. So it's involved in cell cycle arrest, apoptosis, senescence, mitochondrial oxidative phosphorylation, making ATP and NAD our primary source of energy, and electron receptors, glutamine metabolism, glycolysis regulation, and glycolysis is of course the one that cancer cells like the most, lipid metabolism, antioxidant regulation, and homeostasis of cell metabolism, and redox balance. All these functions are potentially compromised by antibiotic P53 knockdown. So it's P53 is located on chromosome 17. It's the guardian of the genome. There are other tumor suppressor genes as well. And so here it shows the good things it does and how it gets rid of bad things. There are stressors. Antibiotics create reactive oxygen in mitochondria and poison those organelles. Turning my attention to pesticide chemical residues because they do most of the mutations that complete the cancers. I have a little bit of addendum on that as well coming up. Methylation, alkylation are also important. DNA methyltransferase damage. Diet. Water, antibiotics, pesticides, lotions, creams, cosmetics, they can all do DNA damage and lead to formation of free radicals. Pesticides in China. Okay, their effect on the mitochondria and ubiquitin proteasome system in Parkinson's disease. Why is this relative? Well, the ubiquitin system is really a signaling system that controls developments in translation, and we need to know more about what the antibiotics do to that. But individual pesticides have been found to cause mitochondrial dysfunction and ubiquitin proteasome system dysfunction. These are some of the pesticide-induced cancers. Okay, these have been linked to pesticides, all of these cancers. So pesticide exposure is an important alternate backup potential carcinogen, which can modify outcomes and increase the risk. This is a mechanism for aging and cancer, errors of replication, blockage of transcription, reduced gene expression, mutations conferring selective advantage for clonal expansion, pre-malignant field defects, and functional decline of tissue and organs. Mutated P53 has many effects, and these are some of them. Inhibition of DDR, apoptosis, autophagy, glucose lipid metabolism, glucose import, oxidative stress response, permissive microenvironment. These are all things that can happen and be affected by protein folding. Types of DNA damage, double-strand, single-strand breaks, these are common in mitochondrial damage, and adduct formation as well. Fusobacterium impairment leads to enhancing expression of pro-inflammatory cytokines via ROS, and this can lead to harm. So P53 gene has also been affected by the seniors in our society who got vaccinated for polio and Hep B. Some of them may have got SV40 monkey virus at that time, and that has been linked to cancer, in particular mesothelioma. So there's also another Jekyll and Hyde personality mutated P53. It's called Puma. P53 upregulated modulator of apoptosis. In this case, the cancer cell actually takes the P53 gene and makes it its protector. So this has to be looked for as well. I talked about microbiota protocancer influences already. Now, microRNA have been found downregulated in breast cancer, and I really think we need to know if antibiotics can do this. And so we need to get involved in looking at the microRNA and their silencing, their methylation, or their upregulation in cancer and in relation to drugs that we give to patients. Also, there's mutual regulation of microRNA and DNA methylation. They play an important role in cancer initiation, progression, and metastases. And Wang and Wu have done a great job on this, looking at microRNA. And we need to examine that more. For regulatory approvals, these coding and non-coding genes have all been silenced epigenetically in cancer, and we need to know what is silencing them. And certainly we need to know if antibiotics are capable of silencing them. And so we should be looking to see if these things are silent after antibiotic administration. This will give us a good clue as to what cancers are being caused, because these are all mapped back to different cancers. Now, when I looked at pesticides, I found similar effects on mitochondria. 9 common pesticides tested induced morphological changes to mitochondria at low concentrations. Paraquat, chlorpyrifos, pendimethyl endosulfan peroxamate, and tibupirad induced mitochondrial fragmentation. So knocking out the oxidative phosphorylation. That gives cancer a leg up. Paraquat, rotenone, chlorpyrifos, peroxamate, and tibupirad cause significant dose-dependent decrease of intracellular ATP, suggesting increased risk of Warburg syndrome because ATP is a proxy for oxidative phosphorylation integrity. Proteasome integrity also disrupted, and we need to look at health consequences. So do antibiotics cause cancer? Do they induce mitochondrial malfunction, damage, or rupture? Yes. Prevent mitochondria from fatal reproduction in quality and quantity? Yes. Cause mutations, genetic material damage? Yes. Interfere with tumor suppressor genes? Yes. Create microbiome dysbiosis? Yes. Harm the immune system? Yes. Increase the Warburg effect, increase dominance of glycolysis over oxidative phosphorylation? Yes. And are there statistically significant association? Yes. Now, the extent and degree of that for any particular antibiotic hasn't been proven, and we need to go and do research on that. But overall, as a class of substances, there is sufficient evidence to suggest that these are cancer contributing. So the evidence that I've looked at means that statistical evidence exists for prostate, breast, lung, colon, ovary, endocrine, liver, non-melanoma, skin, duodenum, pancreas, kidney, bladder, male genitals, thyroid, myeloma, leukemia, bowel cancer, fatal breast cancer. Genetic evidence, smoking pistol, hepatocellular carcinoma, and the P53 mutation at locus 249. Reactive oxygen, lipid peroxide, mitochondrial, other mutations, and immune depression, and Warburg implicated in many cancers. These effects are most likely contributory to other cancer promotional events in a person's life. So I have a theory on how this all comes about. The antibiotics produce cancer, carcinogenic mitochondrial reactive oxygen and carcinogenic mitochondrial lipid peroxide, and reduce anticarcinogenic mitochondrial glutathione. They're absorbent and cause DNA mutations. Antibiotics mutate P53 tumor suppressor genes and possibly other tumor suppressor genes, or possibly silence P53, or increasing risk of carcinogenic mutations. Loss, mutations, and ruptures of mitochondria probably results in loss of oxidative phosphorylation and ATP metabolism, increasing glycolysis favored by cancer, i.e. the Warburg effect. Microbiome dysbiosis leads to the loss of short-chain fatty acids and immune depression, promoting carcinogenic infectious agent environment. They may lead to reverse Warburg, where P53 becomes a carcinogenic agent, cancer agent, supported by epidemiology, and P53 mutation at locus 249 causes HCC. So, there's suggestions ubiquitin proteasome integrity affected by reactive oxygen, leading to oxidative stress, cell aging, and cell death. This might facilitate carcinogenesis associated with aging, although that last one is not so certain. So we need to research the safety of antibiotics, drugs, and pesticides on mitochondria, carcinogenicity, and cardiovascular systems. Vitamin C very happily absorbs reactive oxygen, and E absorbs lipid peroxide. So there's a simple way to tackle this right off the bat. Possible antioxidant administration, N-acetyl-L-cysteine, and bacteriostatic antibiotics, but we need to test the safety and efficacy. I have some research proposals resulting from this work, and you can also find my draft article at the site in the bottom here. How large and persistent, these are my suggestions. How large and persistent are glutathione decrease and increases in reactive oxygen and lipid peroxide per antibiotic? What effect is the effect of all antibiotics on genome, structural, and reproductive integrity of coronary mitochondria? Survey and list genetic mutations, silencing in cancer genes, and key microRNA. Duration and detail of microbiome dysbiosis and short-chain fatty acids and other immune actions of each antibiotic. Assess coronary and mitochondrial health after antibiotic treatment. Antibiotics classigenicity for each antibiotic at application concentrations. Test antibiotic rupture and reproductive integrity of mitochondria per antibiotic. Assay oxo-DG post-antibiotics and ATP post-antibiotics for carcinogenicity potential from loss of oxyphos. Assay tumor suppressor genes P53, TUSC, and TPMS for upregulation, downregulation, silencing mutations at their loci post-ABS. Assay BC genes post-ABS for epigenetic silencings, that's breast cancer. Look for Warburg effect, increasing glycolysis with decrease of OP. Antibiotics, red meat, and ethanol history, epidemiology for HPV-linked oropharyngeal and CRC cancers in men in developed countries. Assay post-ABS for reverse Warburg by H1F alpha and FOS-Jun activity. Assay ABS treatment for caveoli-1 and adjacent fibroblasts, which increases mitophagy and lactate driving tumor growth. Survey for inflammation post-ABS because resistant pathogens may have been activated. Warning, antibiotic resistance in the plant and animal world protects beneficial microbes as well as pathogens, and critical functions like oxidative phosphorylation, ATP, and all those organisms that use mitochondria for energy and NAD for electron transfer. Let us tread carefully in creating more powerful antibiotic so that we do not mess with this global gift of evolution and create another existential crisis.